Our last lecture on organic molecules deals with nucleic acids. These are the molecules that are found that make up things like DNA and RNA, which I'm sure you've heard of. So nucleic acids are basically long chains of nucleotides joined together via the process of, you've heard it before, dehydration synthesis. Okay, so when I join two or more nucleotides together, I'm creating a nucleic acid. There are three parts to a nucleotide that you need to know. All nucleotides have a sugar group, and that sugar group can be something like deoxyribose. Okay, OSE means that it's a sugar. It could be ribose. Oops. Or it can be whatever you want it to be, depending. Okay, so every nucleotide has a sugar group. Every nucleotide has a phosphate group, which you should remember from our discussion of functional groups. You should remember what that is. And lastly, every nucleotide has a nitrogenous base. You need to know that. The sugar group, the phosphate group, the nitrogenous base. There are different nitrogenous bases. I'm going to talk about those on the next slide. I'll give you a couple of examples of nucleic acids. DNA is one example of nucleic acid. It's a nucleic acid where I have a bunch of nucleotides joined together. Each of those nucleotides has as its sugar group deoxyribose. By comparison, RNA is another nucleic acid which consists of lots of individual nucleotides joined together, but in those nucleotides their sugar group is ribose. ATP is a third type of nucleic acid that I'll talk about in just a couple more slides. This is the energy storage molecule for our cells. Okay, now let's take a look at the structure of DNA very briefly. And I want to mention here that in a few weeks we're going to spend an entire chapter looking more in depth at the structure of DNA, how DNA copies itself, and what DNA is used for inside of cells. For today, we're simply looking at the chemical structure of DNA. DNA is a molecule that consists of two long chains of nucleotides joined together to make a nucleic acid, and those two chains spiral around each other to form a double helix. Again, remember that the sugar group found in the nucleotides is deoxyribose, and those nucleotides are joined together once again by dehydration synthesis. Okay, DNA has a sugar phosphate backbone. Okay, the sugar phosphate groups are along the backbone, and the nitrogenous bases are in the center, like a ladder. Remember I told you before that a nucleotide has three parts, a sugar group and a phosphate group. Those make up the backbone of the DNA, and the nitrogenous bases are found in the center. Those nitrogenous bases join up with each other. And there are different types of nitrogenous bases. There's one group of nitrogenous bases that are called the purines, and they consist of two rings of carbons, and they include adenine, or the letter A for short, and guanine, or the letter G for short. We also have what are called pyrimidines. Pyrimidines are a single circle or ring of carbons, and they include cytosine, or C, thymine, or T. Those are all in DNA. In RNA, we also find a different pyrimidine called uracil, or U, and we find that in place of thymine, but we'll talk more about that later. Now, these purines and pyrimidines pair up with each other in a highly specific manner, such that adenine will always only pair with thymine, and cytosine will always only pair with guanine. And when they pair up together, they pair up by forming hydrogen bonds between them down the center of this ladder. If you have a hard time remembering who pairs with whom, just remember that the letter C is made of curves and the letter G is made of curves, and they pair together. The letter A and the letter T are all made with straight lines, and so the straight line letters pair together. Again, this is just the very tip of the iceberg on DNA and RNA. Later on in a few chapters, we will go into much greater detail on these molecules. Last thing I want to, oh, I'm sorry, I got one more slide before I get to ATP. Let's talk briefly about RNA. Whereas DNA was a double-stranded helical molecule, RNA is single-stranded. And in its nucleotide, instead of having deoxyribose as the sugar, like we did in DNA, here we have ribose as our sugar. We still have a sugar phosphate backbone, as you can see here, with the nitrogenous bases sticking out to one side. The one special thing to remember about RNA is we do not have thymine in RNA. Instead, we have uracil. So we still have adenine, we still have guanine, we still have cytosine, but we don't have thymine, we have uracil instead. This chart right here gives you a good idea of some of the basic differences between DNA and RNA that you should know and understand. Things like what type of sugar group is found in the nucleotides of each, what bases are found, because the 4 bases found in DNA are different from the 4 bases that are found in RNA. That DNA is a double-stranded molecule, whereas RNA is single-stranded, that DNA coils to form a helix, and that RNA does not. You should be comfortable with those facts and understanding what they mean. Lastly, I want to very briefly talk to you about what ATP is. ATP is a type of nucleic acid. It is a nucleotide just like we see individual nucleotides in DNA and RNA. That means it's composed of a sugar group, a phosphate group, and a base. The base is adenine, and instead of just having one phosphate group, here we have 3. So it's not adenine, I'm sorry, it's adenosine. Okay, yes, it is adenine. Here's my adenine. Here is my sugar group. And then instead of having one phosphate like we see in DNA and RNA, we have 3 phosphates bonded to it, which is why it's called adenosine triphosphate. What's special about adenosine triphosphate is that the bond here between these 2 phosphates is highly unstable, and it tends to break apart very readily, and when it does, when this breaks apart, energy gets released. And we use that, we store energy in the form of these ATP molecules, and then when we're ready to perform work inside of cell, we break apart that very unstable bond, releasing a phosphate group, and then we take the energy that we derive from breaking apart that molecule and use it to perform work inside the cell. All right, that's it for nucleic acids. That's the end of our tour of organic molecules and the end of your required content for this week.